Okay, so I'm going to be going through the steps of DNA replication in this video. So DNA, you all know, has a double helix. Okay, and so the first step that happens in DNA replication is that the helicase itself is unwound. Okay, or you could say it's unzipped. You're allowed to say that as well. And this is done by the enzyme DNA helicase. All right, there are 3 enzymes that you need to know when you go through DNA replication. So the first one is that DNA helicase unzips, and you are allowed to use that word. I put it in quotes. Oh God, I hope you can see this. But the idea is that, so the DNA helicase unzips the DNA molecule. And basically what it's doing when it does that is that it's breaking hydrogen bonds between the nitrogenous bases. All right. So for example, here, if you have an A over here, you would have a T. Let's try. And then if you have, I don't know, a C over here, you would have a G. And so basically it's separating those two. Why is this not focusing better? All right, and I'm going to underline the enzymes so that you have those. So DNA helicase is the first one. And then the second thing that happens is you get nucleotides that come in and match these bases. All right, so free nucleotides. And you want to be clear on how you're phrasing this. Okay, so the idea is that the free nucleotides come and pair up with the exposed bases. And I either mention the idea that this is via the complementary base pairing rule, or, and I always do both, you would literally mention like A with T, C with G. Okay, so free nucleotides come and pair up with the exposed bases via the complementary base pairing rule. And the other thing that you can mention, so I already mentioned that the hydrogen bonds are broken between the bases, but if you wanted to say that they are binding via hydrogen bonds, you could do that as well, okay? So again, A always goes with T, C always goes with G. For this type of question, if you're being asked about a large, like it's a 4 or 5 point question, you're asked to describe, they usually do accept the abbreviations. Again, if you want to be super cautious, then you could write out adenine and thymine and cytosine and guanine, but usually just A and A, the letters are acceptable, okay? The other thing is that for this, when you're talking about it, you want to always mention that this is happening on both strands of DNA. And the reason I'm stressing this is because this is one of the key differences between this and for transcription. And so if they ask you to compare and contrast DNA replication with transcription, this is one of the big things. So this happens on both strands of the DNA. Remember, again, we're being very careful about how we're talking about this, right? This whole thing, the double helix, is a DNA molecule, but each DNA molecule has 2 DNA strands. So the strands are the individual, each one has 1 phosphodiester backbone. All right, so it's on both strands of the DNA. Okay, and I'm going to pause it and try to draw some out here so that you don't have to watch them. Okay, so I've got a few more bases here written down, and the idea is that the free nucleotides that come in, so they're going to come in on both sides. So for example, here you would have a nucleotide with a T, here another T, here you'd have one with an A, and here you would have one with a G. Okay, and on the other side, the same thing. You'd have one with an A, well, the complementary thing, I should say. One with an A, one with a T, one with a C, and another one with a C. Okay, so for example, these are the free nucleotides that are entering. Good Lord. And that's happening simultaneously on both sides of the DNA as it's being spread open by the DNA helicase. All right. So then what you have is you have your second enzyme come in. And so the idea is that the DNA polymerase actually helps this happen. It helps the lining up, and it also creates the bonds. It adds on the individual nucleotides. Okay, so DNA polymerase, which I'll underline. So it's going to catalyze the lining up of the nucleotides and the connection between them, okay? And so the idea is that you need to talk, when you talk about these mononucleotides being added on, then the idea is that you need to name, again, name the bonds that are happening. Always make sure you're including the name of the bonds, okay? And so the idea is that the free nucleotides that are being linked together, the bonds that are forming between them are phosphodiester bonds. So these are phosphodiester bonds. Your book says something slightly different, and it doesn't talk about phosphodiester bonds with DNA polymerase, but when I get to the third enzyme, I'll talk about, they actually both are forming phosphodiester bonds. Okay. And then, so then you have the phosphodiester bonds, and then the last step, oops, sorry, is that, I'm going to write that over here. Then you have DNAligase, which is the third enzyme that is involved in DNA replication that you guys should know about. So DNAligase, that's a G. Okay, now it looks like a weird Q. Anyway, so the DNAligase, but what it does is it links together pieces of DNA, like the longer stretches of DNA. And that is also, it also creates phosphodiester bonds. The difference between these two, and your book doesn't go into this, but just so you understand, is that DNA polymerase can only add individual nucleotides to a growing polynucleotide strand, whereas DNAligase is capable of linking together long strands, like two long strands, two polynucleotides together. So the idea is that if you have 6 here and 6 here, DNA polymerase is not capable of linking those two together, okay, whereas DNAligase can. So it links together the polynucleotide strands. I'm being a little more particular on this than the mark schemes need you to be, all right, because of my own background in this. And so the idea is that this links together the polynucleotide. Let me say, no, it's fine. And so this is also done, forming phosphodiester bonds. And if you're really concerned about this, don't be, because on mark schemes, they do not particularly care. As long as you have one of these enzymes mentioned in the correct location, they don't really stress about which one of these is forming the phosphodiester bonds. As long as you have a correctly named enzyme, that's usually worth a point. Okay. And so what I mean is that the idea is that the DNA polymerase, for example, would be adding on the individual nucleotides as it goes up this way, whereas the DNAligase would be adding the bigger strands here. So if you have a strand of these 4 and a strand of these 4, then it's going to be able to connect those two. It has to do with the directionality of the DNA, which is not something other than the fact that you do need to understand that the two strands, when they're opposite each other, are anti-parallel. Okay, and that has to do with the idea that if you have your mononucleotide here, right? So this is a cartoon of a mononucleotide. And you have your phosphate, your sugar, and your nitrogenous base. When these get connected together, right? So I'm drawing a second mononucleotide here. And so this is the phosphodiester bond that's formed by DNA polymerase if you're adding on a single one. But if you remember, right, the idea is that when these are linked up, the nitrogenous bases are anti-parallel. I'm going to try to draw this. You guys know I'm an artist. There we go. It looks like this. Right, so you can see the point of the sugar is going in 2 different directions. All right, so there's a directionality to these strands that these enzymes care about, but your mark scheme doesn't. So that's why that's happening. Yeah, and then it connects like this. Okay, so the idea is that they're headed in opposite directions, and these enzymes are limited by that direction. But if you're asked to describe DNA replication, this will always get you the points, okay? Make sure you're naming the types of bonds. Make sure you are very clear about the idea of free nucleotides coming in and both strands being used as template strands. And so the end result then, if you wanted to tie it all together, the idea is the end result is that now you have 2 molecules of DNA, and each one has 1 new strand and 1 old strand. Old strand and 1 new. And the other thing, of course, and so of course, this whole thing is semi-conservative, and you want to make sure you mention that because that is also often worth a point, right? And I've already done, you can check out the other videos on how we know it's semi-conservative, the Michelson install experiment on what's going on with that, okay? So those are your 3 enzymes. Don't forget, you can always lead with this or you can end with it. You can say when the 2, the idea that 1 old strand and 1 new is semi-conservative, or you can start out by saying DNA replication semi-conservative and then go through the process. It doesn't matter how you do it, as long as you're logical about it. All right. That's it for that one.